sir. Good afternoon, good morning, good evening, sir. Happy holidays to you, sir. Sir, we are the Group 8 and we would report the Laboratory 8, the DNA isolation from strawberries. So, introduction. In 1953, James Watson and Francis Crick proposed a molecular model of DNA, or deoxyribonucleic acid, after a year and a half of joint work at Cambridge University. So completely was their model of substantiated by subsequent investigation. The team shared a Nobel Prize in 1962 with Maurice Wilkins. The basic unit of structure of the DNA molecule is the nucleotide. This is also the basic unit incorporated in DNA during synthesis. The nucleotide consists of three parts. So those three parts are phosphate, sugar, and nitrogenous bases. So next. So this is the objectives of laboratory 8. First is to extract deoxyribonucleic acid using the materials given. Second is to separate the deoxyribonucleic acid from the strawberries. So for this laboratory, these are the materials given. So 1 to 3 strawberries, about the volume of a golf ball. Frozen strawberries should be thawed and at the room temperature. Then milliliter DNA extraction buffer, soapy salty water, about 20 ml, ice cold 91% or 100% isopropyl alcohol. First, one Ziploc PM bag, three test tubes, test tube brush, test tube holder, test tube wrap, one funnel, one coffee stirrer or transfer pipette, 10 grams of baking soda or 2 grams of sodium chloride or salt, microscope glass slide, cover slip, triple beam balance, and 1 to 10 ml graduated cylinder, 2 water bath, white cloth, and a toothpick. Next, what is DNA? DNA is present in every cell of plants and animals. The DNA found in strawberry cells can be extracted using common everyday materials. Strawberries are soft and easy to pulverize. Strawberries have a large genomes. They are octoploid, which means they have 8 each type of chromosomes in each cell. So next, these are the materials in pictures. First is strawberries, alcohol, and beaker, and a white cloth, and a team bag, dish soap, cylinder, and tweezers. So procedure for Rovelin Pimentel.
So, the procedure in performing the experiment: first, remove the green sepals from the strawberries. Second, place strawberries into a Ziploc tin bag and seal shut. Third, squish for a few minutes to completely squash the fruit. Fourth, add the tin emulsion extraction buffer or the soapy salty water and squish for a few minutes. And try not to make a lot of soap bubbles. Buffer must be chilled, placed within ice. Soft buffer solution: 120 ml of water and 5 grams of sodium chloride and 5 grams of baking soda. Fifth, filter through a moistened paper towel set in a funnel and collect the liquid in a clear test tube. Do not squeeze the paper towel. Let about 5 ml of liquid. Sixth, add the alcohol gently to the top of the solution, about an inch and a half above the solution. Next, add 2 volumes ice-cold isopropyl alcohol to the strawberry liquid in the tube. Pour the isopropyl alcohol carefully down the side of the tube so that it forms a separate layer on the top of the strawberries liquid. 8. Watch for about a minute. What do you see? You should see a white fluffy cloud at the interface between the two liquids. That's DNA. 9. Spin interface between the two liquids. Joke. 9. Spin and steer the coffee sear or transfer pipette in the tangle of DNA, wrapping the DNA around the sear. Then pull out the sear and transfer the DNA to a piece of saran wrap or clean tube. The fibers are thousands and millions of DNA strands. To view in a microscope, put the glove on a clean slide and gently tease or stretch apart using two toothpicks or dissecting pins. The fiber will be easier to see the tease apart area. 12. Rinse your funnel. Put the vacuum bag in white cloth in the garbage. So in the first pictures, the squished strawberries, put the TM bag in with the sodium chloride or the salt. Second, filter the squished strawberries in a moistened paper towel set in a funnel. Third, gently add the alcohol to the extracted strawberries. We saw the alcohol separated from the strawberries. And the fourth one, the fluffy cloud we see on the surface of a toothpick was the strawberries DNA. Next. So the results and discussion. Were you able to isolate the DNA fragment from your sample? Well, yes, the hydrophobic tail attached to the squished strawberry cells that pulls the membrane away from DNA. The cloudiness above in the DNA fragment and below is extracted strawberries. So, for the continuation, let's hear it from Ms. Sampson.
So in the table below, you can see the purine bases that needs to be paired with pyrimidine bases. So there are two types of purine bases, which is the adenine and guanine, while also there are two types of pyrimidine bases, which is thymine and cytosine. So the purine bases, adenine is always paired with pyrimidine thymine, and the pyrimidine cytosine is always paired also with the purine guanine. So this interaction is known as hydrogen bonding, like A is like to pair with T and C is with G. Next. So this is the structure formula of the DNA molecule that is properly labeled its three components, which is the phosphate group, nitrogen base, and the sugar. Next. So to determine it, covalent band is the sort of band that hyperlink among nitrogen base pairs, sugar, and phosphate organization. Adenine, guanine, thymine, cytosine, and uracil are the five not unusual place nitrogenous bases. So covalent bands shape among the phosphate organization of one nucleotide and the 0.33 carbon atom of the pentose sugar within the side of the following nucleotide. So each strand is composed of nucleotides bonding together covalently. So they connect together through their both paired groups to form these long polymer chains which are the bases of DNA. So the order of the subunits is extremely important, and it is how the genetic information is stored. It gets a little more complicated though, because then it isn't just one chain, it is actually two, and they are held together by interactions between the nitrogenous bases. Next. So last will be the conclusion. So our conclusion is that all of the materials we employed are significant in the experiment because the smashing of strawberries physically breaks the cell wall. DNA extraction buffer aids in the release of DNA from the cell and nuclear membranes, whereas salt rejects negative DNA charges, reinforcing DNA strands and precipitating proteins and carbohydrates. Ice cold isopropyl alcohol also pulls water from DNA molecules, causing them to collapse and precipitate. DNA appears as white fluffy cloud strand particles, and as we remove the mucus stuff from the beaker, we see that as the alcohol evaporates, the white fluffy cloud substance solidifies, indicating that our DNA is extremely robust and cannot be easily damaged. So, as a result, we can confidently state that we have successfully isolated DNA from strawberries, and also that when you added the salt and detergent mixture to the smashed strawberries, the detergent helped lyse the strawberry cells, releasing the DNA into solution, whereas the salt helped create an environment where the different DNA strands could gather and clump, making it easier for you to see them. A single strand of DNA clumped in this activity, you will be able to see just how much of it three strawberries have when all of their octoploid cells are combined.
So we have questions ready for you. So these questions are well made, and we have to answer it because this of recorded session. So first question: what steps did we use to extract the DNA? First, we broke apart cells by physically squishing the fruit. So, Robelin, please answer it.
So the chemical detergent process broke down the cell walls, the cell membrane, and nuclear membranes. The fruit mixture was cooled to stop the DNA as released from the cytoplasm from the destroying the cell's DNA. The mixture was filtered to separate out the large cell parts that are not needed. The DNA was the precipitated through chemical means the ethanol.
So, thank you, Robelin. So, we proceed to the second question. Explain what happened in the final step when you added ethanol to your strawberry extract. Miss Sheila Salusan, please answer
So,
it.
since DNA is not soluble in ethanol, it separated itself from the strawberry extract and rose to the top of the test tube when we added this alcohol in the final step. That's all. So I'm gonna also question you. And the first question is for Mr. Retiza. So what did the DNA look like? So relate what you know about the chemical structure of DNA to what you observed today.
The DNA looked like white thin fibers in a clump. Because DNA comes in a long strands, I was able to identify the DNA that each individual strand was a piece of DNA,
So
deoxyribonucleic acid.
okay. So Rovelin. So what was the purpose of smashing up the strawberry?
Well, to break down the cell wall, cellular and nuclear membranes to open the cells.
Okay. Next.
So here's the question for you, Miss Samson. So why are strawberries used for DNA extraction?
So ready strawberries are a great hotspot for separating DNA since they are anything but difficult to pummel and contains proteins called pectinases and cellulitis that helps them to separate the cell dividers.
Okay, so for you, Mr. Retiza, is strawberry DNA the same as human DNA?
Um, Human cells have two duplicates of their DNA. Strawberry cells, then again, have eight, which means octoploid. This means make it simpler for us to separate DNA to have the option to see it with unaided eye at the end. It is not it is very small. That's all.
Okay.
Okay.
So
So
this is the laboratory 8 of group 8. I hope you will understand the reporting, sir, and also happy holidays, sir.
happy holidays, sir.
Happy
Happy holidays,
holidays.
sir. Thank you,
Thank
sir.
you.
Thank you.
Bye-bye, sir. Love you. Okay, love you mo.
I love you, sir.
And hi.
That's all, sir. Thank you.
Thank you po.
Pag-like mo, pag-like. Camera.